Hello everyone, I am Deepti Mishra, Assistant Professor in Modern Institute of Pharmaceutical Sciences, Indore. Today I am going to discuss about urea cycle in biochemistry. So, the urea cycle is the first metabolic pathway and is also known as Krebs-Hanslitt urea cycle. It is also called ornithine cycle because ornithine is the first member of the reaction. So, urea is synthesized in the liver and transported to the kidney for the excretion in urine. The 2-nitrogen which is involved in the urea is derived from the different sources, one of the ammonia and the other directly from the amino group of aspartic acid. And in urea, the carbon atom is supplied from the CO2. So, urea is the end product of the protein metabolism. Urea accounts for 80 to 90% of the nitrogen containing substance excreted in urine. So, urea synthesized in basically five-step cyclic process with the 5 distinct enzymes. The first 2 enzymes which are present in the mitochondria and the rest of the localized in cytosol. This is the urea cycle, and in this cycle ammonia is condensed with the CO2 and form the carbomyl phosphate, and carbomyl phosphate is react with the ornithine and form citrulline. Citrulline is converted to the arginosuccinate and then arginosuccinate is converted to arginine and release the urea and regenerated the ornithine. So, in detail we can discuss. First step is formation of carbomyl phosphate. First, carbomyl phosphate synthesis, this is the enzyme of mitochondria, catalyze the condensation of ammonia ion and CO2 to form carbomyl phosphate, and this step consumes 2 ATP. This is the step of formation of carbomyl phosphate. First is ammonia is condensed with the CO2 with the 2 AP in the presence of carbomyl phosphate synthetase and form the carbomyl phosphate. Second step is formation of citrulline. The second reaction is also in mitochondria. So, citrulline is synthesized from the carbomyl phosphate and ornithine by the ornithine transcarbamylase. Ornithine regenerated is and used in the urea cycle. So, ornithine and citrulline are the basic amino acid. Citrulline is transported to cytosol because of the two steps are basically two steps are involved in mitochondria and rest of the steps are involved in cytosol. So, citrulline is neither present in the tissue protein nor in blood, but it is present in the milk. This is the second step, formation of citrulline. Ornithine plus carbomyl phosphate in the presence of ornithine transcarbamylase form the citrulline. Third step is the formation of arginosuccinate. So, citrulline is condensed with the aspartate to form arginosuccinate by the presence of enzyme arginosuccinate synthetase. The second amino group of the urea is incorporated by the aspartic acid. It requires 2 ATP, it is cleaved to AMP. The fourth step is formation of arginine or cleave is the arginosuccinate. So, enzyme the arginosuccinase or arginosuccinate lyase responsible for the cleaving. So, the arginosuccinate lyase cleave the arginosuccinate to arginine and fumarate provide the arginine is continue the process and the fumarate provide the connecting link with the TCA cycle or gluconeogenesis. So, fumarate is converted to the oxaloacetate via the fumarase enzyme and aspartate is regenerated in this reaction. This is the last step, this is formation of urea. So, arginase is the fifth and final enzyme that cleave the arginine to yield the urea and or regenerate the ornithine. So, ornithine is regenerated, enter the mitochondria for its reuse the urea cycle and arginase is activated by these coenzymes. So, ornithine and lysine compete with the arginine. Arginase is mostly found in the liver while the rest of the enzyme of the urea cycle are also present in other tissue. So, arginine synthesis may occur in varying degree in many tissue. This is the urea cycle. In this cycle, basically 2 steps are processed in mitochondria and other steps are processed in cytosols. First, carbomyl phosphate synthesis, ornithine transcarbamylase, arginase, arginosuccinase synthetase. They are the basically 5 enzymes which is involved in the urea cycle, and this is the 5 basic steps in urea cycle. Thank you all.